to you.
Okay, thank you. So I'll just try to share my screen. And if you let me know that that comes through, okay.
Will do.
I have to let it share. How does that look?
That looks great.
Okay, great. So thank you everyone for giving me the opportunity to give you this talk. Today I'm going to talk about our self-amplifying RNA technology, and then tell you about how we have developed that, how we have made the coronavirus vaccine and what we targeted, and then show you the data from our animal studies. And also at the end, I'm going to mention a few other bits that we do in relation to vaccine hesitancy work. So I just start by explaining what are nucleic acid vaccines. So you've probably all heard about DNA vaccines, and in the news recently, of course, the Pfizer and the Moderna vaccines have highlighted that there is the other category of nucleic acid vaccines, which are RNA vaccines. Now, these are not new technologies. These are technologies that were developed in the 1990s and refined over the next 10, 15 years. And they actually went into clinical trials. I'm talking here about RNA vaccines. They went into clinical trials as early as 2013. So that was for a rabies vaccine, and that was a company called CureVac that did that phase 1 trial. And it showed that these vaccines were safe, that they produced antibodies against the rabies glycoprotein, and that they could potentially be used as a way of vaccinating the general population. So whenever people say things like, oh, we have no safety data from these vaccines, this class of vaccines, we actually do. We have safety data from 2013. There have been 7 or 8 phase 1 and 2 clinical trials with different proteins and different companies, mostly CureVac and Moderna, it has to be said. But there is long-term safety data there. So whenever people mention that, oh, there's short-term safety from the coronavirus vaccines, there is actually long-term safety from the other trials. There is another class of therapeutic, it's called Onpattro, and it's a therapy which is targeted towards the liver and is given intravenously. And it is essentially a piece of RNA that is lipid encapsulated and is almost identical to the lipids used in the Pfizer BioNTech vaccine. So that shows that there's already also, not only is there safety data, but there is a licensed medicine with an RNA lipid entity. So back to nucleic acid vaccines. The target can be expressed from either a DNA or RNA template, and you know this just codes for the protein that we want to make. So you all know that the normal biology is DNA is in the nucleus. It makes an RNA strand which moves to the cytoplasm, and it's there that it produces the protein, which is the target for a vaccine. And it goes in that method, in that orientation. So the RNA cannot go back in biology in mammals to DNA. And of course, protein cannot go back to RNA. So why do we want to use RNA vaccines? What are the advantages here? So if we look at this schematic, we can see that the top bar shows the safety of a potential vaccine, whereas the bottom bar shows the potential efficacy of a vaccine. And this is actually a slightly outdated schematic, which doesn't really take into account the fact that adjuvants have been able to dramatically enhance the efficacy of things like subunits and virus-like particles, as well as nucleic acid vaccines. Now, there is no adjuvants in the current batch of RNA vaccines, but in the future, we could potentially add those in. If we look at the idea of safety, obviously a live attenuated virus is potentially the most unsafe mechanism to administer a vaccine, as there's potential for it to almost de-attenuate to become more wild type, become more vigorous. With the nucleic acid vaccines, the safety profile is thought to be excellent, and that is actually being what we're seeing in the current clinical trials. So the other advantages of the vaccines as nucleic acids are speed of development. They're much easier to modify than, say, even a replicating vector or a virus-like particle. They're very cheap to make. They can encode virtually any proteinaceous antigen. They're safer, and they have, because they're made in the body that we want them to be expressed in, they have the adequate and suitable post-translational modification that would be the same type of modifications that would happen if it was a virus making it inside your body. One of the other advantages is that it can express membrane-bound proteins. A lot of the other proteins, like particularly for subunits, they have to be made so that they're soluble, so that they can be purified and made to GMP. So, self-amplifying RNA vaccines are slightly different than plasmid DNA and even different from messenger RNA. So the messenger RNA and the self-amplifying RNA do not require penetration into the nucleus. So as well as not being able in biology to go back into DNA, there is no risk of any kind of integration of the RNA into the host genome. When we look at the schematic here, we can see the messenger RNA, a typical messenger RNA strand is about 2,000 nucleotides, whereas a plasmid DNA, of course, is much larger, is 7,000 base pairs. But also one of the disadvantages of self-amplifying RNA is its size. So in comparison to a messenger RNA, it is 9,500 to approximately 12,000 nucleotides in length, depending on the particular replicon structure you use, and also depending on the insert size. So with it being much bigger, it is much more difficult to deliver, and that has been one of the major roadblocks for self-amplifying RNA. But it's something that we have been working on for a number of years and have partially solved. But also, the advantage of why people use the saRNA instead of the messenger RNA is the concept that getting in even a small amount of the self-amplifying RNA will allow the amplification process to make multiple copies of the antigen of choice, of the vaccine target antigen. Whereas the messenger RNA, of course, will last for an amount of time in the cell and will produce a quantity of the protein and then will disappear. Now, you'll know that the two vaccines that are being promoted at the moment, the Pfizer-BioNTech one and the Moderna one, are modified messenger RNA types and not self-amplifying RNA. So for our particular replicon, we used a Venezuelan equine encephalitis virus as the replicase backbone. So here you can see that the NSP1 to 4, which are the non-structural proteins from the genome of that virus, have been separated away from the virus. So that's the only components of the virus we use. There's a 5' UTR, a subgenomic promoter, and also a 3' UTR. And whenever we express from this replicon, we are able to express the non-structural proteins 1 to 4. And this is the replicase complex. This acts on the replicon from the 3' end. It retranscribes the entire replicon and makes a minus strand. So this minus strand is then able to remake itself in with the same non-structural proteins 1 to 4. I won't go into the mechanism of the process of the NSP1 to 4 because it's different combinations of those 4 proteins that make the different replicase units that do all of these different functions. So it's quite handy that there is actually a way that the virus has developed to make this and that we have used, but the actual NSP1, 2, and 3, and NSP1, 2, and 3 plus 4 is quite complicated, and I don't have time to go into all of that today. So then we decided that we would, in January, start the process of designing a coronavirus vaccine. So what would we need to do? And where did the coronavirus come from? We all know about the SARS-CoV-1 virus, which is the one that had an outbreak in 2003. And we know that that outbreak was almost self-limiting in that it infected quite a large number of people, but then seemed to disappear. But there's also 5 other coronaviruses that infect humans, in addition to the MERS, which is the other severe respiratory one. The human coronavirus NL63, 229E, OC43, and HKU1, these are all our common cold viruses. And they circulate routinely through people each winter. 10 to 20% of us will catch one of these viruses. But apart from the human coronaviruses, there are many others that infect animals. And what they do, which is quite interesting actually, is that they have a complex method of generating new viruses. Not only do they mutate, but they also have major recombinations between viruses. So for example, if this coronavirus is moving through several hosts, it would mutate gradually to be more adapted to that host. But also, if there was 2 coronaviruses genomes that are present in that one host, then it could make a completely new variant by this technique called RNA polymerase switching. And they switches between genomes, so it copies one part of the genome, then switches to the other genome and makes a completely recombined variant. And it's these that are generally the ones that are successful in jumping to new species. So with COVID-19, this is thought what happened in probably from a bat virus that then was in a wildlife market, and they were not sure exactly which species it was in. People say pangolin, but others say it was some kind of a stoat type animal. But they're not entirely sure in that wildlife market what it was circulating through, but that's how the COVID-19 emerged. How do we know what protein is the one that we should focus on? So you can see, like in this schematic, there is a very prominent S protein, and then the membrane protein and the envelope protein, which are tiny proteins that make up part of the outer capsid. And we knew from the SARS outbreak in 2003 that it was the S protein that contained the receptor binding domain for cells, human cells, and they bound to the ACE2 receptor. So we knew that this was the target that we should look for our vaccine. When the Chinese scientists released the information on the genome, we were able to see quite quickly and easily where exactly the S protein was. So we can see it here. And if we then look at that sequence, and this was done very early on with the novel coronavirus, we can see the structure of the receptor binding domain, and we can see the orientation of the 3 chains which make up the spike protein. And here in the middle of this, you can see some EM that we did on our protein. And you can almost, they're not great pictures, but you can almost see the structure of the spike protein. This was a soluble one that we made initially. And as I said before, this is how the spike protein binds to the surface of human cells through this ACE2 receptor. So all of that was known, and all of that was research that was done since 2003. So we had all of this information, and you can say that for this particular disease, we were well ahead of the game. We didn't have to work out exactly what protein would be the target. We didn't have to worry about how it works. We could from the SARS coronavirus 1, we could easily map what exactly was needed to be done for a vaccine. So the decision to make the vaccine was based on having all of this prior knowledge. So as I said, our vaccine is a strand of RNA. You can see here the actual RNA on a gel, a reducing gel. And the length of our particular replicon was quite large. It was 11.5 thousand base pair. And whenever we put that into the cell, we are able to detect the product of the subgenomic promoter, which is many copies of the SARS coronavirus spike protein. So what else did we need then to make the RNA vaccine against coronavirus? Well, not only, of course, do we need the spike protein in the replicon, but we also need a way to deliver it, a way to get it into the animals. What we used in this case, because we had been working on delivering different vaccines, well, for example, we had developed vaccines against Ebola, Lassa, and Marburg. We had used lots of different formulations, different polymers, different ways to try and make it enter the cell. And we had come up with, before the coronavirus started, we come up with the using lipid nanoparticles. These are a collection of lipids, and a ionizable lipid is one of them, which enables us to encapsulate the RNA, deliver the RNA to the cell. The cell then takes up the whole lipid particle, and because of the ionizable lipid, when it gets into the late endosome and the change in pH happens in the late endosome, it is able to rupture the endosome and allow easy entry of the RNA into the cell. And prior to coronavirus, we had found that this was the most effective way that we found to get significant quantities of the vaccine RNA into the cell. So based on that earlier research, we decided to do that for this also. So we formulated it with lipid nanoparticles, and then we started the first mouse experiments to confirm that the RNA that we made was able to make a spike protein that had some immunogenicity. What was our decision timeline for making the vaccine? The circulating strain was sequenced early on by the Chinese scientists on the 10th of January. We accessed the sequence by the 19th of January. We had designed the entire what we were going to make by the 21st of January. And we assembled the genes by the end of the month and confirmed expression of the saRNA vaccine of the spike protein from the vaccine. And then we started the immunogenicity studies on the 13th of February. We also, with in mind the fact that we would be wanting to take this into the clinic. I mean, we were being overly ambitious at that time, probably in our own heads, I think. And we decided we would start a toxicology study in rats, which is a necessary prerequisite for any kind of going into first in humans. So we started that by the end of March. And we had submitted our paper in April, and we had started our clinical trials in June. So you can see that for us, the timeline of making the vaccine was actually extremely short. Making the vaccine, it was less than a month. Then the rest of it has to happen. The rest of the animal studies, all of the paperwork, all of the getting clinical trials organized, and then performing the clinical trials. And essentially that's what we've been doing since then. So because we're still in the phase 1/2 part of our human clinical trial, I can't share any of that data with you, unfortunately, because of course it's still blinded. But I can talk about the immunogenicity in the mice and what that has as a potential impact for human results. Is it working? So surprisingly to us, the vaccine actually worked extremely well. So the doses here, if you look at the very right-hand side, the left-hand side of the graph, you can see that this is our previous best type of vaccination, which was plasma DNA with electroporation. And we could see there that the amount of antibodies raised was very high. You can see 10 to the 5 nanograms per mil of specific antibody. And also on the very right-hand side of this graph, if you look at the recovered COVID-19 patients, you can see the spread of the amount of antibodies that are in those patients. And even at doses of 0.01 microgram of formulated, LNP formulated self-amplifying RNA, we were achieving levels that were higher than any of those that we'd previously seen. So we were quite excited about this, bearing in mind, of course, that it's mice and not people. But we thought that the potential dose sparing and the immunogenicity of this particular spike protein in the context of an saRNA vaccine was high. So that encouraged us to continue further, and that's probably when we decided to initiate the toxicology studies. We also, of course, looked at the neutralizing potential of the sera from those mice. And again, if you compare it with the DNA on the very left-hand side, you can see the neutralizing potential is 10 to the 3 IC50 units. And also on the very right-hand side, we've got the recovered COVID-19 patients. So the recovered COVID-19 patients actually were particularly useful for all of these studies. And the reasons for that are quite interesting scientifically, in that a lot of different labs do their analyses in slightly different way. They have set up their own ELISAs, they have set up their own pseudovirus neutralizations, they have set up their own wild type neutralization assays. And it's very difficult to cross compare between labs. Even if they do a very similar protocol, you can get slightly different results. So having the recovered COVID-19 patients was critical here for us to be able to benchmark our results against everyone else's results. And of course, everybody was doing the same thing. So you'll notice in many COVID papers that there's always recovered COVID-19 patients as a spread. So if we look at this pseudovirus neutralization, so if I just explain the pseudovirus neutralization, it's essentially a particle which contains luciferase as an enzyme, and that particle has got the spike protein on the surface. So whenever the particle is put onto target cells, if you pre-incubate that with antibody that blocks the ability of the spike protein to bind to those cells, then you will get a much lower signal from the luciferase. So what we can see here is the potential to neutralize. With our 10 micrograms and 1 microgram, you can see that there's a very high neutralization score here after the second shot. So the shots here were given at 0. So at week 4 here, we can see the results after 1 shot of the vaccine. And at week 6, that's 2 weeks after the second shot. So you can see the potential for neutralization goes up massively. So we were quite buoyed up by these results also. But pseudovirus neutralization is also a difficult measure. And what you want to do is actually a wild type neutralization. So a wild type neutralization is where you have the actual virus and you're looking at the ability to reduce the plaque titer of the virus on a cell monolayer. So, but again, we saw here that the LNP vaccines had a very high neutralization titer in the wild type neutralization assay. This is seen as the essentially the gold standard. And when we correlated the levels of the antibody against the spike protein, with neutralization, you can see there is a really excellent R-squared value. So the higher the amount of antibodies, the higher the neutralization titer. And this is pretty much what we've seen for all of these studies, and it's also what everyone else has seen, that the neutralization, unlike something like HIV, where it's actually difficult to generate neutralization titers which are able to prevent infection, and it's also does not usually correlate with the levels of antibody. So for HIV, you can generate huge amounts of antibodies against HIV, but very seldom are any of them potently neutralizing. For this particular protein, it appears that if you generate any antibodies at all, you will generate antibodies against the RBD domain, and the RBD domain, the receptor binding domain, is where the neutralization occurs. So you have a good neutralization score. We also looked in mice at different aspects of the antibody response. So a T helper 1 response is one that is supposed to be good for producing high affinity antibodies. In these particular mice, it's always skewed towards a Th2 response. So with our vaccine, we were able to skew it back towards a Th1 response. That was also positive news, but not necessarily indicative of what would happen in people. But one of the other things that we did see, which was quite heartening for this particular vaccine, and also something that other people have seen with this protein, is that with the saRNA vaccine, we can see extremely high levels of T-cell responses. And these are measured in this assay in the splenocytes of the mice, and it's interferon gamma T-cell responses after stimulation with specific peptides. So if we look again at the plasmid DNA, which is typically a good stimulator of T-cell responses, you can see here that the levels are 6 to 7 times higher than the plasmid DNA. The reasons for this are unclear. The plasmid DNA is given in these mice with electroporation, but I don't think it's just a result of the saRNA itself. I think it's probably a result of the delivery method. The delivery method of LNPs, I think, is able to produce robust T-cell responses. So it's also an observation that was noted in the other RNA trials, which were not saRNA, but which were modified RNA. So, and I've changed this slide actually a number of times already, because the clinical trials take longer than you would think. So we originally had started in June, and we were hoping for our phase 1 and 2 to be finished by end of October. So we think that's end of December now, because we had to do several modifications of the dose that we were giving. And we're still hoping that we can start our efficacy studies, which are the phase 3 trials, by the sometime in January of next year. And of course, they will take a length of time to complete. For all of the trials, everyone has found that there are the volunteer uptake has been very high. So with volunteers willing to come forward, it makes things move quicker. Usually with our clinical trials, we're desperately trying to recruit, and that's what takes most of the time. But our recruitment is not a problem for the coronavirus vaccines. I wonder, actually, will it be a problem going forward whenever everybody is actually being vaccinated? Let's hope not. So we're hoping our efficacy studies will start in January, and we will be recruiting large numbers of people, probably more than 5,000, but it depends on whether we go down, which route we go down for the efficacy study and what's required. And then the implementation is in the future. It says May to June there, but that's probably being optimistic also. So I would hope that we would be able to have our efficacy studies finished by the middle of next year. people may be asking why we're continuing now that there's already vaccines, and I think that the answer to that would be there's lots of people that need to be vaccinated. There's lots of people making these vaccines, and they all seem to be pretty successful. I would imagine that the demand for the vaccine will be continuous for the next year at least, and therefore the more people that make it, the better. We hope that our efficacy would be as high as what other people have seen, so any vaccine should be good. And the safety profile of RNA vaccines, I think, will compare extremely favorably to the safety profile of any other vaccine. So from during this process, what kind of lessons did we learn? So the lessons that we learned mostly were probably a little bit about big pharma compared to academic labs, in that obviously big pharma can move mountains quite easily, but even for the coronavirus vaccine, we had to secure significant funding and significant support as an academic outfit to move forward. So I think for me, the lesson might be that academics probably aren't the best placed people to push a vaccine through to an efficacy trial, and that it will probably be necessary to have some kind of collaboration and agreement with a medium or large pharma enterprise in order to have a vaccine go through to the final stages. Now, this is something that we probably knew intellectually already, but it's not something that in practice we'd ever done, because we'd only ever done phase 1 trials, so which we could do very successfully, and we did many of them. But going from phase 1 to a phase 3 is an entirely different thing, and it's where big pharma really comes into its own. But other lessons learned about how we make the vaccines, we know that the Oxford group are making the adenovirus vaccine, and they're actually doing this really successfully, even though they have to produce extremely high titers of that adenovirus per dose, because the dose is 5 × 10⁻⁻10 viral particles for a single vaccination. But for viral vaccines in general, you obviously need extremely large bioreactors. And this is where Oxford had the advantage and the experience, in that they had already set up, way back in probably February, March, they had set up their capacity because they had a good idea that their vaccine would work well. They set up their capacity to be able to make large quantities of this. But of course, it's a big industrial effort. And if we look at the history of making vaccines, we can say that for example, for flu in the 50s, people were making flu vaccines in eggs. In the 2020s, they're still making them in eggs. We need large bioreactors for the viral vaccines, which you can solve, but it's expensive. It's a large industrial type unit. But for RNA vaccines, we can make 100 mils would be at least 1 million doses, which I think is something to take away from this, in that the RNA vaccines are far more efficient in being able to manipulate the vaccine, to change the vaccine, especially in response to a pandemic, but also in the production of that vaccine. The scale up is probably a lot easier to do. And I think that's one of the reasons why the two front runners for this have been BioNTech, Pfizer-BioNTech, and also Moderna, because they have really got this process down to a T, and they're able to make large quantities of doses quite quickly. So whenever we started this endeavor, we were thinking, how do we make 5 million doses? We didn't know what our scale would be, but we had made a plan, and our plan was to have the RNA made in a company in San Diego. We took it to a company in Vancouver, and got it formulated with the lipids. And then we took it to different places to then get it vialed. I mean, it's things like that that you don't realize become, roadblocks, is that the vialing process to GMP is also extremely difficult to do. And then, of course, we sent it back to the UK for our clinical trials. So for the future, and this is something where I think that RNA vaccines can come into their own. In the future, we want to have some kind of distributed effort of making the vaccines, making them in those small bioreactors in different places. So we would hope that instead of just having it in a few small numbers of places throughout the world, that these small bioreactors can be moved to different areas of the world and provide the access, which is becoming increasingly clear that that access is not available for many of the countries listed on this schematic. So you can see that while Europe and America is well supplied with vaccines at the moment, if you look at well, actually not all of Europe, if you look at the vaccines that can be potentially supplied to other parts of the world, it's still low numbers. We need to have some kind of way of distributed vaccine so that these countries can make the vaccine themselves and distribute it to their own populations. So I finally want to talk about other work that we do is because we're very invested in vaccines, we have partnered with the UN and the Vaccine Confidence Project at London School of Hygiene and Tropical Medicine. And we've started this online social media campaign where we are talking about vaccine hesitancy and trying to counter the arguments and the conspiracy theories put forward by the anti-vax movement. The anti-vax movement has become, like most other social media phenomena, has become very organized and is really becoming a fringe movement of its own. And of course, the anti-vaxxers don't speak for the majority of people. The majority of people sometimes express vaccine hesitancy, and vaccine hesitancy is completely different from anti-vax ideology. Vaccine hesitancy is simply people saying, oh, this seems to have been made within a couple of months. You're asking me to inject it into my body. Can you guarantee me that it's safe? And I just want to ask you a few questions about that. And you can see that being vaccine hesitant is a perfectly normal response, especially to a vaccine that has been made in such a speedy manner. And here again, there's a lesson for the scientific community in that we made a big deal of saying, oh, we've done this really quickly. And of course, the scientists who are able to analyze the data and look at the antibody responses and the safety profiles and the inflammatory response, etc., that's all fine. But if you say to a general member of the public, even somebody who's highly educated but in a different field, you say, oh, we did this really quickly, aren't we great? And of course they just go, oh yeah, and you want to inject that into me, okay? Is it safe? And those questions are perfectly okay. And what we're trying to do with this Team Halo project is to try and address those questions, try to online answer questions that people have about vaccines and whether we can guarantee that they're safe, or we can tell them what the ingredients are, we can tell them whether there's eggs in them, all of those things. And so we do this on various platforms, one of which is TikTok. And TikTok is, we decided to do that one because it was geared at a very certain demographic. I'm not sure it's been terribly successful in reaching a demographic. It's certainly been quite successful at getting anti-vaxxers out and shouting about things. But so we basically make TikToks to try and explain different aspects of the vaccine and to answer questions. But we also do other things like Reddit's Ask Me Anythings, all those kinds of things. So another thing I wanted to share with everyone here is that there is this excellent online app. So it's a web app, and it was designed by Ed Parker and Beata Kampman, who used to be Imperial and now are at London School of Hygiene and Tropical Medicine. And this is an app that has all of the clinical trials that are happening throughout the world, what stage they're at. It's got links to each one, and it's got all the criteria as to the inclusion, the number of participants, all of those things. So it's a absolutely fantastic app for seeing what's happening in the COVID vaccine world. So the conclusions from our work is that the saRNA vaccine is highly immunogenic. It produces high levels of antibody and neutralization antibodies, and it's similar or better than the natural infection in recovered patients. Our phase 1 is set to begin. It did begin, and our future challenges are the modular manufacturing of RNA. And with that, I want to acknowledge the people who have been part of this work. It's been funded by Innovate UK, CEPI, EPSRC, and Precision Nanosystems. And at Imperial College, all those people have been involved in the work at Imperial. The University of Bristol helped us with the protein characterization work, and Acuitas Therapeutics in Vancouver are the people who helped us to prepare the LNP and formulate the saRNA. And with that, I'll open myself to questions.
Thank you so much for that, Paul. I think I speak for everyone when I say that was just a fantastic talk. So just to remind everybody, if you have any questions, you can ask them in the Q&A box, or you can raise your hand and I can unmute you. So we already have a few questions, so I'll go through them in order. So Georgia Perona-Wright, who is also my postdoc supervisor, has asked, you mentioned that the lipid nanoparticle delivery system is especially good for getting strong T-cell responses. Do you know why this is, and is it related to the types of cells that the lipid nanoparticles deliver the RNA into?
Yeah, so I don't know why it is. I suspect that it has something to do with the ability of the lipid nanoparticles to get to the draining lymph node. So this is a comparison to something like plasmid DNA, which of course transfects all, when you give an intramuscular injection, it transfects the myocytes and you produce high levels of protein, which are then taken to the draining lymph node. But I think that and we are actually working on this and not just us, but lots of people, to try and resolve how the LNPs work, because we know that they do get into, of course, they do get into the muscle cells also. But because we're actually injecting fantastic quantities of LNPs, I think that they drain very easily to the lymph node. And because of the size of the particle, they're more able to drain to the lymph node. And I think that they're able to then directly transfect different cells than you would normally see in the muscle. So maybe follicular dendritic cells, and this is where the investigations need to go. We need to look and see exactly what cells are being transfected. So it's exactly what you say. I think it's the different types of cells.
That's really cool. Thank you. So Maro Gaia is asking, well, saying thank you a lot for the great talk and all of your efforts, and has a question about how do you stop the self-amplification of the RNA vaccine?
Yeah, so, well, we don't stop it. It kind of stops itself. Like any other biological process, it has an ability to be wound down by protective mechanisms in the cell. So if you put it into the cell, it usually requires some kind of sequestration, and we see this in, it's called spherules, and they're like little, on the cell membrane, they're little like sort of invaginations of the membrane and the RNA preferentially replicates in there because it's being protected from being sensed because it goes through stages, of course, of being a double-stranded RNA, which we all know for most cells is highly inflammatory. So saRNA does set off these inflammatory signals, and that's probably why it is a self-limiting reaction. I think that whenever we have, we also have done this test where we have got, this is going to be a bit gross, we have got skin from elective surgeries, mostly stomach reduction and or breast reduction. So we get the extra skin from that. And of course, that at that point is non-vital tissue. But whenever we have injected intradermally into that skin, we have seen the saRNA last for 2 to 3 weeks. And then it does go away. But again, this is non-vital tissue. And we would love to know what actually does happen in vivo. But I think with it may be a bit of a non sequitur actually, because non-vital tissue, it might last longer. I think that when in vital tissue, it's got much more mechanisms that are trying to get rid of it. So as I say, with this double-stranded RNA phase of the replication of the replicon, I think that's highly inflammatory, and that's what limits the saRNA. Of course, it can't become incorporated into the DNA, so that's always a positive thing. And it also doesn't seem to then travel to new cells. So, each cell is constantly giving off little microvesicles of itself, and it doesn't seem to travel in those to new cells.
That's really interesting.
And highly speculative.
But still, it's a really interesting. So the next question is from Jonathan Urbach, who says, "Thanks so much, very interesting talk." And he's wondering, how immunogenic are the NSPs, and what is the relative expression of NSPs and spike?
So the NSPs, we have looked at it, and they are immunogenic. There is a response, but they're very minimally immunogenic. I can't really answer as to why. I think it's because they go through degradation. This is one of the things I was talking about during my talk, is that the whole process of the NSP1 to 4, it's quite a complicated complex. So it goes to an NSP1 to 3, and then the 4 adds on, comes off, adds on again. There's protease digestion of the 1, 2, and 3, and each different aspect of that protein actually has a different structural conformation and does a different function. So I think that some of those are extremely short-lived, and if they were longer-lived, then we would have issues with it not replicating properly. So I think because it gets made and then degraded very quickly, the actual target for things is fleeting. And also, of course, that might mean that the final degradation product of the NSP1 to 4s, which we probably never look for, do exist there, and probably might be relatively immunogenic, but because there's so many protease sensitive sites in that polyprotein that I think the end one might be immunogenic, but because we don't even know what that is, I would imagine that I've never looked at it, and I don't know who else has.
Okay, great. And yeah, there's a question from Vinod Ilangavan, and they ask, well, it says, fantastic talk, and ask, will it be possible to use nucleic acid aptamers in the future as a quick therapeutic agent?
Yes, so that's quite a large study in its own. Aptamers and ribozymes were mooted quite a few years ago as being potential therapeutic agents. And they suffer from a very similar problem as RNA, in that it's the delivery of these things that becomes the critical roadblock, rather than designing an aptamer or a ribozyme that's able to do a function, because all of those things can be designed quite easily to a function, but it's getting them to enough cells to have an appreciable biological effect. Maybe with LNPs that would be improved, but certainly, of course, aptamers work very well, and there's many of them now.
Oh, that's a really interesting thing to think about. So, okay, well, if no one has any more questions for now, would you be able to put your Twitter handle in the chat box so that people can make a note of it if they have additional follow-up questions?
Sure. Where is the chat box?
It should just be at the bottom. Should just say chat.
Chat. So what's my Twitter?
So yeah, I mean, so thank you very much again for that talk. I think like I said before, speak for everyone. I think is really is fascinating, and obviously even more fascinating because of everything that's going on at the moment. So thank you for taking the time to come and speak to us about that. And also, I'd just like to say thank you to everybody who has attended again. This was the last seminar of the year. We've got another couple coming up in January. But in the meantime, I hope you all have a very lovely, relaxing holiday through December, and hopefully see you all again in the new year. So